Okay, the final class of large biological molecules, nucleic acids. So, ultimately, the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. So our genes are kind of like recipes. Like each gene is a recipe for a certain thing. And so basically the recipe is kind of telling you, okay, put this amino acid and then this amino acid and then this one, and then you're going to fold like this, and then boom, you make this hemoglobin. So yeah, nucleic acids do have like a direct connection to proteins in this way. The genes end up telling you how to make a certain protein. So the amino acid sequence of a polypeptide is basically told by a gene. Genes consist of DNA, a nucleic acid that is made of monomers called nucleotides. So yeah, nucleic acids are polymers, and their monomers are called nucleotides. And there are two kinds of nucleic acids, DNA and RNA, and so we'll talk about them. DNA stands for deoxyribonucleic acid, so it does tell you in the name, nucleic acid. And then RNA is ribonucleic acid. So these are both types of nucleic acids, and they have different roles, but they both play a part ultimately in like gene expression. So DNA gives directions for its own replication, and we'll talk about that when we get to the cell and how cells divide. DNA has to also be divided, like be copied so that there is enough to be divided into two cells. But DNA also helps direct synthesis of RNA. So there's a special kind of RNA called messenger RNA or mRNA. There's actually multiple kinds of RNA. So DNA directs synthesis of messenger RNA, and through the mRNA ends up controlling protein synthesis. So again, building protein. And so all this process is actually what gene expression is. So this whole class, we're going to talk about all these different things, and then ultimately about how genes are expressed. And again, that ties into the big picture, right? Like you are who you are because of your genes and how they get expressed. I think this picture got cut off, but you know, we're going to go into more detail in this in a future unit in the class. But yeah, DNA is kind of like a recipe, and sometimes you don't want to have the whole cookbook in the kitchen because if it gets damaged then you have to buy a new cookbook or whatever. I know there's phones and you can look up recipes, but you know. In this old school analogy example, DNA is a recipe, and so you copy down a recipe card in the form of RNA, and then you take that little recipe card and then that can leave the nucleus of the cell, and then that could go to the kitchen, that's the cytoplasm, and then cook up protein. So, you know, we're gonna go into way more detail about this. Don't worry about it too much, but that's how DNA and protein kind of come together in this way. Protein, the DNA tells the mRNA how to be made, basically, and then the mRNA tells the cell how to build proteins. So each gene along a DNA molecule directs synthesis of a messenger RNA. That messenger RNA molecule interacts with the cell's protein-making machinery to direct how to make a polypeptide. And this flow of genetic information can be summarized like that. So DNA to RNA to protein. Okay, so yeah, all that's kind of just previewing, foreshadowing, all that. But let's get back to nucleic acids. So they are polymers made of monomers called nucleotides. So you connect one to build the whole polymer. One nucleotide has a nitrogenous base, a pentose sugar, remember carbs, and then one or more phosphate groups. So those three parts make one nucleotide, and then you connect one nucleotide to the next to build a nucleic acid. And so these can kind of vary, the nitrogenous base. The pentose sugar can vary depending on if it's DNA or RNA. And then yeah, there will be one or more phosphate groups. So the whole thing is a nucleotide. The part of a nucleotide without that phosphate group is called a nucleoside. So a nucleoside is just that nitrogenous base and the sugar. There are two families of nitrogenous bases. There are the pyrimidines, which include cytosine, thymine, and uracil. And so the pyrimidines, even though they're different, they're kind of grouped together because they have a 6-membered ring, kind of like shown here, 1, 2, 3, 4, 5, 6. So this is kind of like a pyrimidine example. And then the purines consist of adenine and guanine, and this has a 6-membered ring fused to a 5-membered ring. So adenine and guanine have that kind of backbone in common, and then vary slightly. So those are the different nitrogenous bases that could be there. Cytosine, thymine, uracil, adenine, or guanine. And in DNA, that sugar right here, the pentose, is either deoxyribose, or in RNA, it's just ribose. So the nucleoside is just the base and the sugar, and then a nucleotide is that plus a phosphate group. I feel like we're not really going to talk much about nucleosides. We're mostly just going to talk about nucleotides, so the whole thing. So this is kind of putting all that stuff together. So this is one polynucleotide, or nucleic acid, again because it's a polymer. So one nucleotide connected to the next, and then yeah, the rings can have different rings of the nitrogenous bases there. The sugar can be different. So here on the right, on the top, are the different bases. The pyrimidines just have the 6-part sugar. Purines have the 6-part sugar connected to a 5-ring. Sorry, the 6-ring. It's been a long time of recording already, okay? So purines have 2 rings, the 6-part and then the 6-membered. And then the sugar is either ribose in RNA, and so here overall you see this shape, and then at this part right here there's an oxygen, so that's ribose if it has the oxygen. If it doesn't have the oxygen, it's deoxyribose. So that's what makes DNA and RNA different, or one of the things, is that the ribose has that oxygen, deoxyribose has this sugar. So again, even the smallest change in chemistry makes a totally different molecule. Yeah, okay, so let's wrap this up. So nucleotides are linked together by a phosphodiester linkage to build a polynucleotide, or nucleic acid. A phosphodiester linkage is made of a phosphate group linked to the sugars of 2 nucleotides. So the phosphate group linked to the sugars of 2 nucleotides. And these links create that backbone of sugar phosphate units. So all this purple is like the backbone, blue I guess. And then what sticks out are the bases. So this is like what's represented basically when we show the DNA double strand, like the strand is this backbone. And then the links that look like the ladder coming together are the bases connecting on the opposite strands. So this is what I mean. This strand is just kind of showing that backbone, sugar phosphate backbone, and then what sticks out in these pictures are representing those rings. So it's not that accurate, right? And that sequence of what bases are there, of those nitrogenous bases, is what's unique for each gene. So in total, a DNA molecule has 2 strands connected to each other, spiraling around an imaginary axis, forming that double helix. And those backbones actually run in opposite directions, and so we call that anti-parallel. So if this end has a phosphate group sticking out and this end has the hydroxyl group sticking out, the opposite one is the other way. The phosphate is on this side and then the hydroxyl is on this side. And so one DNA molecule is super long and it includes many genes. And so when those 2 ladders and strands are coming together and sticking out, only certain bases will be able to pair up and form hydrogen bonds to do this. So if you see how green and yellow are always together, wait a second, this is not accurate. So adenine always binds with thymine, and guanine always binds with cytosine. And so this is called complementary base pairing. It's because spatially what sticks out, these are the only things that are compatible to bond to each other. Adenine always binds with thymine, guanine always binds with cytosine. So this is complementary base pairing. And this feature of DNA actually makes it possible to kind of like unzip and then build identical copies of each DNA molecule when the cell's ready to divide. Okay, so that was DNA. And so RNA is super similar. The only differences are, the ribose, and one difference here. So RNA is single-stranded, and because it's single-stranded, sometimes it could actually kind of base pair with itself. So if it were to kind of fold, it could fold with itself and interact like that. So complementary pairing can happen between 2 RNA molecules or between parts of the same molecule. In RNA, thymine can't really hang out there for whatever reason. That's just how it evolved. So in RNA, there's uracil instead of thymine. And so A and U will always pair if there is RNA involved. And DNA is always a double helix, but RNA molecules can vary because of it being single-stranded and how it can bond to other things or itself. So yeah, DNA, double-stranded helix. RNA can bond to itself or other RNAs. And there are multiple kinds of RNA, so you'll also hear that throughout, the course. Okay, so this is a summary for nucleic acids. A nucleotide has these 3 parts: a nitrogenous base, a sugar, and a phosphate. And then it depends on what kind of nucleotide it is, or nucleic acid, if it's DNA or if it's RNA, what the sugar is, what the bases can be, and if it's double-stranded. Alright, so let's wrap this up. So lastly, biologists study, the sequence of DNA, and then also the proteins that get built, because it's all relevant. So once the structure of DNA and its relationship to amino acid sequence was understood, biologists wanted to decode the genes by learning, like, okay, if these are the bases, then what does this mean? What is this going to code for? What kind of protein is it going to make? And so the first chemical techniques for DNA sequencing were developed in the 1970s and refined over the next 20 years. So that made it easier, more efficient, and cheaper to sequence DNA and figure out what is the sequence of nucleotides. And it is enlightening to sequence the full complement of DNA in an organism's genome. So I kind of mentioned that I worked with the model organism Caenorhabditis elegans, these tiny little roundworms. And so because they're small especially, they were able to sequence all of the genes, pretty much all the genes, in these worms. And so we knew what almost every gene coded for and what it was going to make. So it's super interesting. And the rapid development of faster and less expensive methods of sequencing was a side effect of the Human Genome Project. Scientists wanted to figure out, what is the human genome? What are all the genes coding for? So since then, many genomes have been sequenced, generating large sets of data. And so yeah, now there's automated DNA sequencing machines. It's not as old school as it was when we first learned. And so there are new fields. So bioinformatics uses computer software and other computational tools to deal with the data resulting from sequencing genomes. And analyzing large sets of genes or comparing whole genomes of different species to each other is called genomics. And if you're studying large sets of proteins and their sequences, this is called proteomics. And so if we study the DNA sequences that are kind of common to different species and different groups, we can use DNA and proteins as kind of like tape measures of evolution. So the sequences of genes and their protein products help document the hereditary background of an organism. Especially because these linear sequences of DNA are directly passed on, basically, from parents to offspring. So we could kind of extend the concept of genealogy to molecules and to relationships between species. So molecular biology has another measure to the toolkit of evolutionary biology this way. Okay, so that is everything. That is lecture 3, large biological macromolecules, and this is a big picture summary again. A lot of these are polymers made of monomers. There are carbohydrates, proteins, nucleic acids, and lipids. Yeah, so that is this lecture. These are kind of the main ideas. And these are the learning objectives. What you can hopefully do now at the end of studying all this, watching the lectures, hopefully still studying, reviewing the lecture notes, maybe reading the textbook, maybe watching additional videos. These are things that you can hopefully do, by the time a quiz or exam comes up. Okay, that's it.